Breast cancer is an extremely prevalent form of cancer, which affects 1 in 8 women across the United States in at least one point of her lifetime. In an effort to combat this devastating disease, researchers and doctors have identified 3 receptors that can broaden the treatment types for the patient: the estrogen receptor, the progesterone receptor, and the HER2/neu receptor. When a pathology report is done, the tests done on these receptors all come back negative in the case of triple-negative breast cancer. This narrows the treatment options for the patient, as the treatment cannot target these specific receptors. Younger women, African American women, and women with mutations in the breast cancer 1 gene are more susceptible to triple-negative breast cancer. Triple-negative breast cancer is typically harder to detect on a mammogram and, compared to other types, tends to grow more aggressively. Until recently, not much had been found in terms of genetic susceptibility for breast cancer, triple-negative in particular.
In determining a population of genomes to analyze, researchers found that while most families severely affected by breast cancer did not carry mutations in any of the typical susceptibility genes, founder mutations were disproportionately present in the sector of the Finnish population affected by breast cancer. The prevalence of those known mutations indicated a strong probability that the affected Finnish population would also carry other mutations and susceptibility genes.
To perform the study, the team collected data from 24 breast cancer patients from 11 Finnish families using exome sequencing. Some selected variants were genotyped in additional cancer patients and controls. In Phase 1, 524 BRCA1/2 negative patients. In Phase 2, 552 population controls. And in Phase 3, 1,730 unselected breast cancer patients, 233 familial BRCA1/2 negative patients, additional to those in Phase 1, and a total of 1,274 population controls, including the 552 controls from Phase 2 from the Helsinki region, as well as 679 unselected breast cancer patients, including 257 familial cases, and 816 population controls from the Tampere region of Finland were genotyped. The purpose of testing in different phases was to compare the DNA sequences of triple-negative breast cancer patients, controls, and breast cancer patients. Variants were filtered so that only those that were predicted to change protein sequence were considered. They were then filtered again to include only those that were predicted to be pathogenic. The team then manually verified the remaining variants to verify their structures and positions.
In attempting to determine causes for higher rates of breast cancer in Finland, the team looked at certain genetic mutations and their prevalences. They noticed that women in Finland had specific gene mutations which made them prime candidates for genetic testing to identify if there are new genes that cause cancer predisposition that haven't already been identified. Their main focus was DNA repair genes, and they identified that a prime suspect was the FANCM gene with the mutation P.Q1701X as the cause for higher predisposition to triple-negative breast cancer, or TNBC. This mutation was found in 2.9% of breast cancer patients and 5.6% of TNBC patients, but only 1.4% of controls, all living in the Helsinki area. This mutation makes women 1.86 times more likely to get breast cancer and 3.56 times more likely for TNBC. Another part of the group's focus was that family history played a part in whether breast cancer forms and what type. The P.Q1701X mutation consistently caused elevated rates no matter the familial background. Patients with a strong family history of breast cancer were 3.2 times more likely, but women with only a first-degree relative were 2.9 times more likely. In the Helsinki region alone, patients with a family history of TNBC were 6.33 times more likely to have the mutation. Overall, among unselected TNBC patients, there is a 4.92 times higher chance of the P.Q1701X mutation showing up no matter the familial history.
This study, while mainly focusing on Finnish families, has a wide-reaching effect on the topic of breast cancer worldwide. This is true because our knowledge of the FANCM C.5101CT nonsense mutation continues to progress. We are able to focus on our efforts on what may be the cause of breast cancer, TNBC in particular. While there have yet to be any definitive studies in other areas of the world to precisely estimate FANCM's direct relation with TNBC, it is still possible to use the Finnish study as an initial risk assessment when testing for forms of breast cancer in future patients. This utilization of the case study as a primary risk assessment will not only lead to improvements in prevention, but also early diagnosis and possibly treatment.